So the lac operon is found in bacteria, and it is an operon that is a set of genes that are transcribed or expressed together. The lac operon, the lac in lac operon stands for lactose. And so we as humans also consume lactose. Lactose is a carbohydrate, and it is basically two molecules of glucose that are stuck together. You can see here. So lactose, again, two molecules of glucose that are connected, and in order to use that lactose, then organisms need to break apart those two glucose molecules into two separate glucose molecules so that they could then use the glucose in cellular respiration, fermentation, etc., whatever metabolism they happen to have. Now, as humans, we might also be familiar with the enzyme called lactase. Now, lactase is an enzyme that breaks down lactose, but we're not discussing that particular enzyme here. Lactase is not the enzyme that is encoded for in the lac operon. I know that's a little bit confusing, but the lac in lac operons refers to lactose, not lactase. So if you're lactose intolerant and you take lactase pills, then you're not taking the protein that results from this operon. Instead, the enzyme that breaks down lactose that is made from the genes in this operon is called beta-galactosidase. So again, beta-galactosidase here, we see the spelling along the bottom, or BGAL for short. Beta-galactosidase is the enzyme in this bacterium that is produced, which breaks apart the two parts of lactose into individual glucose molecules. So in other words, when the lactose operon is expressed, beta-galactosidase is manufactured or produced, and that can break down lactose. So we'll go back to this kind of idea of the big picture in the next video. But for now, what I want us to concentrate on are the molecules that are going to bind to this stretch of DNA. So this stretch of DNA, one moment here, is upstream of the gene. So the genes for beta-galactosidase and a couple of other proteins that are expressed when this operon is turned on are downstream of what we're looking at here. So the P is for promoter, and the promoter is always going to be upstream of the genes, meaning that the RNA polymerase is going to start at the promoter and then get to the genes. We can also have an arrangement on a molecule of DNA. There's our RNA polymerase. We could have an arrangement on a molecule of DNA where the genes are on the left and the promoter is on the right. And the RNA polymerase is still going to move from the promoter to the genes. How does this happen? That's because we have two strands of DNA that are anti-parallel, and so we would be reading opposite strands of DNA or alternate strands of DNA in the top scenario versus the bottom scenario here. If you need to, you can go review that in one of my other videos. So again, just a little review, we are talking about the area upstream of the genes. So nothing that we see here in this diagram directly codes for beta-galactosidase. The beta-galactosidase triplets, their codons are downstream of this, so we're not seeing that here. So, next step is what actually binds to the DNA that we are seeing here. Well, since we mentioned our RNA polymerase, why don't we start with that? So our RNA polymerase is a molecule that is necessary to express a gene, because the RNA polymerase is what actually reads the DNA and makes it into RNA, or creates RNA based off of the DNA sequence. And as you know, RNA polymerase in bacteria recognizes the -35 and the -10 sequence along with the help from its sigma factor. So we're not talking about the RNA polymerase finding the sigma factor in this case. They have already attached, they've already bound, and we are just talking about the whole complex binding here at the -35, -10 area. So the RNA polymerase, again, is going to, with the help of sigma factor, bind to the -35, -10 sequences, which is the promoter, and then it should move towards the gene. All right, but before we get too far ahead of ourselves, next we need to talk about what's right next to the RNA polymerase, or what can bind right next to the RNA polymerase, which is the repressor. So the repressor can bind to the operator. The repressor is a protein, and the DNA sequence for the repressor is elsewhere in the genome. It has nothing to do with this set of DNA. It has nothing to do with this operon. It's completely separate. So we have the repressor protein, and it is there. And again, whether or not it is there is not determined by the scenario we're going to talk about. So here's our repressor. Our repressor can bind right here to the operator. So I'm going to show our repressor actually binding to the operator here. We have that here. And our repressor, again, binds right here. It can attach to the DNA. And what do we see here? Well, we see that it is going to actually block the RNA polymerase from moving forward. And the RNA polymerase, if it can't move in this direction downstream, it can't move the other direction, because that's not how the chemistry works. So the gene is not actually going to be transcribed, or rather the operon's not going to be transcribed. So when the repressor is bound, then that prevents everything from moving forward from this point on. However, we can remove the repressor. How do we remove the repressor? Well, it's not us per se, but it's the environment. So we said that the purpose of this whole operon is to break down lactose. So actually, when lactose is present, and I'm just doing a sketch here, again, not the exact chemical structure of lactose, but put an L here. When lactose is present, it will actually attach to the repressor and change the shape of the repressor. So I'm just going to draw the repressor again here, but we're doing a cartoony depiction. It's changed shape. It's no longer flat on the bottom. It's rounded, it's curved, and so it can no longer bind to the DNA. So we'll just make a little bouncy arrow there. So again, if lactose is present, it will stop the repressor from binding. And we'll talk more about this in the next video. All right. And of course, if the repressor is not bound, then RNA polymerase can more easily move forward. Finally, we're going to talk about the cap binding site. Now, what the heck is this? This is not something we've probably heard about before, unless you talked about it in your general biology class. But the cap binding site is where a particular protein can bind. And actually, I need a little bit more space. So the cap protein is unique to certain bacteria, and it will just summarize in it saying that it's part of the cell's system for monitoring how much energy is present in the cell in the form of molecules. So the cap protein also has two different configurations, just like the repressor protein did. And again, the cap protein is not encoded by the genes in this operon. So we have the cap binding protein, and it can be in a form where it binds DNA, or it can be in a form where it does not bind DNA. And the difference here is that when the cap protein is going to bind DNA, it is going to be attached to something called CAMP. Now don't Google this, because Google takes that to be camp, which is not what we're interested in this instance. But CAMP stands for cyclic AMP. And what is AMP? Well, it's related to the molecule that we are already familiar with, which is ATP. ATP, adenosine triphosphate, is a molecule that is said to carry energy or represent energy, be used as energy. And the T stands for tri and the P stands for phosphate. So adenosine triphosphate is what ATP is. So what does that make AMP? Well, that would be adenosine monophosphate. So just one phosphate. And this kind of looks like a D, so, not the best, but this should be a P. So adenosine monophosphate, that can be converted back and forth depending on the organism. AMP can be converted into cyclic AMP and back and forth. Cyclic AMP is just in particular used as a signaling molecule to show, hey, there's a lot of AMP in the cell, and so energy levels are low. We don't have a lot of ATP if we have a lot of AMP usually, because you're using that same adenosine and adding or removing phosphate groups. So when we have cyclic AMP, that can then attach to the cap binding protein. Or maybe a better way to draw this would be to do this. Cyclic AMP attaches to cap, and then when that happens, then we convert this protein into this other configuration so that it can then bind at the cap binding site. And all right, what does that mean? We just talked about a whole bunch of different stuff, two different configurations for this protein. What does it mean when this protein is bound at the cap binding site? So if we have it here, and then we have cyclic AMP, well, what is it right next to? This is right next to our RNA polymerase. And what this complex does is it actually just lends a helping hand to the RNA polymerase. It stabilizes it. It helps it to bind this section of DNA more tightly so that the RNA polymerase can better prepare to be transcribing. That's all. But in other words, having that complex here at the cap binding site means that we're going to get more transcription. There's like a little bit of transcription without this complex, but with the complex, there's much more transcription. All right, so now we've talked about all of the molecules that might bind in this area. And I'm going to stop the video here and talk about the circumstances wherein the beta-galactosidase is actually produced and when it's not produced in the next video.